So today we're going to talk about DNA replication during S phase of the cell cycle. DNA replication begins with a chromosome, and you guys know that a chromosome has two strands of DNA, and those strands are held together by hydrogen bonds between the nitrogen bases. So before any DNA replication can occur and we can get those new strands, firstly we have to unwind the original chromosome and unzip it, kind of like unzipping a zipper. So we're going to unwind and unzip. There are special enzymes inside of your cells that do this. What's happening is that we're breaking those hydrogen bonds that hold the two strands together. After the breaking occurs, we have two single-stranded pieces that are separate from each other. So they've been straightened out. We're going to actually show the base pairs this time. So we have an A, a C, a T, and a G. And because we have two strands of them, we're going to show them separated now. So the A would have paired with a T, the C would have paired with a G, the T would have paired with an A, and the G would have paired with a C. Okay? Now notice that they're not connected anymore, and we have exposed nitrogen bases. After the exposed nitrogen bases are there, what happens is that we get complementary nucleotides coming in, and each of those original strands gets a new strand as its partner. So what happens is we have our same original strands. So we have the first one with an A, then we have a C, then there's a T, and there's a G. We're going to draw our second strand just a little bit further over this time. So our second original strand had a C at the bottom, then an A, then a G, and a T at the top. And what happens is we get each new strand comes in and it bonds and makes those has those complementary base pairs. So the A pairs with the T, the C pairs with the G, the T pairs with an A, and the G pairs with a C. And so now we're going to have those hydrogen bonds that are connecting the old strand and the new strand. The new strand here is represented in red, and the old strand is represented in blue. We're going to do the same thing for the second strand over here. So we have our second old strand, and coming in, we're going to make another strand that's complementary, meaning that the base pairs match up. So T pairs with A, G pairs with C, A pairs with T, and C pairs with G. And again, we'd have those hydrogen bonds being formed between them that would connect the old strand with the new strand. This form of replication is called semi-conservative replication. So I'm just going to zoom in here. Okay. And you can see that each strand is made up of, we have an older one, which is in blue, and a new one, which is in red. And that's really important to recognize, that every new strand of DNA is paired up with an old strand. And that's why it's called semi-conservative, because only part of it is conserved there. So once we're done here, we've added the complementary base pairs and we have two new strands being formed. Now what happens is that our strands, they can actually wind back up again into that twisted double helix. So we're going to draw another arrow here and we're going to show them retwisting up together. So once we're done, we have two new chromosomes. We have an old one with a new strand here, and we have a separate one over here with old and new together. And so these represent the two identical chromosomes. So over here we're going to write two new identical chromosomes. And again, the important term for this is semi-conservative. So semi-conservative replication. And the reason it's called that is because there's half old and half new. So that's what DNA replication looks like if we could draw it out. Those chromosomes that we just drew can then wind up together to make a replicated chromosomes, and they can go on further in the cell cycle. It is important to note that there can be mistakes that can be made during DNA replication. So sometimes within the cell, there's accidentally the wrong base pairs put in there. But there are enzymes that go through and do kind of a proofreading and check for this. So most of those mistakes are fixed before it goes on. So that's DNA replication in a nutshell.